So, so the summary of biomolecules, although there is a be- bedwelling diversity of living organisms, their chemical composition and metabolic reactions appear to be remarkably similar. Okay, there is diversity, but chemical composition and metabolic reactions appear to be remarkably similar. The elemental composition of living tissues and non-living matter appear also to be similar when analyzed qualitatively. So living tissues and non-living matter almost similar qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen and oxygen is higher in living systems when compared to in any in inanimate matter, that is non-living matter. Okay. However, a closer examination reveals that the relative abundance of carbon, hydrogen and oxygen is higher in living systems when compared to inanimate matter. The most abundant chemical in living organisms is water, right? There are thousands of small molecular weight less than 1000 Dalton biomolecules. There are thousands of small molecular weight less than 1000 Dalton biomolecules, amino acids, monosaccharides, disaccharides, sugars, fatty acids, then glycerol, nucleotides, nucleosides, and nitrogen bases are some of the organic compounds seen in the living organisms, right? Now there are 20 types of amino acids and 5 types of nucleotides. Fats and oils are glycerides, in which fatty acids are esterified to glycerol. Fats and oils are glycerides, in which fatty acids are esterified to glycerol. Now phospholipids contain in addition a phosphorylated nitrogenous compound, right? Now only 3 types of macromolecules, that is proteins, nucleic acids and polysaccharides, are found in living systems. Only 3 types of macromolecules, that is proteins, nucleic acids and polysaccharides, are found in living systems. Lipids, because of their association with membranes, separate in the macromolecule fraction. Biolipids, because of their association with membranes, separate in the macromolecule fraction. Now, biomacromolecules are polymers. Biomacromolecules are polymers. They are made of building blocks which are different. Proteins are heteropolymers made up of amino acids. Nucleic acids, RNA and DNA, are composed of nucleotides. Right? Biomacromolecules have a hierarchy of structures, that is primary, secondary, tertiary, and quaternary. Now, nucleic acids serve as genetic material. Polysaccharides are components of cell wall in plants, fungi, and also of the skeleton of the arthropods. Fungi and arthropods, they have the chitin, plants have cellulose. So these are the polysaccharides. They also are they also are storage forms of energy. Example, starch, glycogen in animals and starch in plants. Polysaccharides, right? Proteins serve a variety of cellular functions. Many of them are enzymes. So proteins are enzymes. Some are antibiotics. Some proteins are antibiotics. Some proteins are receptors, and some are hormones, and some others are structural proteins. Collagen is the most abundant protein in animal world, and ribosco ribulose bisphosphate carboxylase oxygenase ribosco is the most abundant protein in the whole of the biosphere. So most abundant protein in the whole of the biosphere is ribosco ribulose bisphosphate carboxylase oxygenase, and collagen is most abundant protein in the animal world. Now enzymes are proteins which catalyze biochemical reactions in the cells. Ribozymes are nucleic acids with catalytic power. This is the exception to the that enzymes are proteins. Not all enzymes are proteins. Some are nucleic acids as well. Examples are ribozymes are nucleic acids with catalytic power. Now proteinaceous enzymes exhibit substrate specificity, require optimum temperature, right? pH for maximal activity. So proteinaceous enzymes exhibit substrate specificity, require optimum temperature and pH for maximal activity. They are denatured at high temperatures. Okay, they are denatured at high temperatures. Enzymes lower activation energy of reactions and enhance greatly the rate of the reactions. The function of enzymes is the lower activation energy of the reactions and enhance greatly the rate of the reactions. Nucleic acids carry hereditary information and are passed on from parental generation to progeny. So nucleic acids carry hereditary information and are passed on from parental generation to progeny. Now exercises. What are macromolecules? They are retained in the acid insoluble pool. Give examples. I told you nucleic acid, polysaccharides and proteins and lipids. What is meant by tertiary structure of proteins? They have the active sites, active pocket site. Proteins have primary, secondary, tertiary and quaternary structure. Quaternary structure example is hemoglobin. Find and write down structures of 10 interesting small molecular weight biomolecules that is less than 100 Dalton. Find if there is any industry which manufactures the compounds by isolation. Find out who are the buyers. Find out and make a list of proteins used as therapeutic agents. Find another applications of proteins, that is cosmetics. Example, explain the composition of triglyceride. So fats and oils are glycerides in which fatty acids are esterified with the glycerol. Okay, so these are the triglyceride, these are the fats and oils. Can you attempt building models of biomolecules using commercially available atomic models? Draw the structure of the amino acid alanine. Alanine, you know, CH3 is the R group. Amino acids, these are the alpha carbon. These are what are the gums made of? Is Fevicol different? Gums, these are the like secondary metabolites. I think Fevicol different. I think it's the same. Find out a qualitative test for proteins, fats, oils, amino acids. Taste any fruit juice, saliva, sweat and urine for them. Find out how much cellulose is made by all the plants in the biosphere and compare it with how much of paper is manufactured by man, and hence what is the consumption of plant material by man annually, what a loss of vegetation. So the plant cell has the cellulose in their wall in the biosphere and they used to manufacture the paper. Okay, describe the important properties of enzymes. So they lower the activation energy, hence enhance the rate of the reactions. Okay, they are proteinaceous. Few are nucleic acids, that is ribozymes. They exhibit substrate specificity, require optimum temperature and pH for maximal activity. So this is it, your chapter.